أعوذ بالله السميع العليم من الشيطان الرجيم من همزه ونفخه ونفثه بسم الله الرحمن الرحيم الحمد لله والصلاة والسلام على رسول الله أما بعد. Welcome back guys to our meiosis class. In our last class we discussed cell division and mitosis, and to serve as a recap, I said cell division is a process by which a cell divides to form new cells. In prokaryotes we have binary fission. In eukaryotes, we have mitosis and meiosis, and I explained a little bit differences between mitosis and meiosis. I said mitosis is for somatic cells, while meiosis is for germ cells. And let's say somatic cells are body cells whose genes will not be passed on to the future generations, while germ cells are cells that are destined to become gametes, either sperm or egg. A cell whose gene can be passed on to the future generation. And then we say mitosis especially is for somatic cell, and mitosis happens in somatic cells, and it is for growth, repair, and healing of damaged tissues, and also as asexual reproduction in some organisms, while meiosis is only restricted to germ cells, and it is for the formation of gametes, which is the sex cells, the egg and the sperm. So we discussed mitosis in our last class, and this class we're going to discuss meiosis. So without wasting much time, we will move straight to meiosis. So just like I explained, meiosis happens in germ cells, and it is for the creation of sex cells, that's the egg and the sperm. So meiosis was first discovered and described in sea urchin eggs in 1876 by Oscar Hertwig, a German biologist. So it was then described the second time, but this time it is the first time at the level of the chromosome by Van Beneden in 1883, who was a Belgian zoologist. So the term meiosis was coined by J.B. Farmer and J.B. Moore in 1905. So meiosis, just like I said, is restricted to germ cells, and unlike mitosis, during meiosis, DNA replicates once, but the nucleus divides twice. So don't forget that. In meiosis, DNA replicates once, but the nucleus divides twice. In female, meiosis is called oogenesis, and it happens in the ovary, while in male, meiosis is called spermatogenesis, and it happens in the testes. In the male, meiosis starts after puberty, while the female, meiosis starts while the child is in the womb. There's at approximately 12th week of fetal development, and become arrested, and it's after it continues in puberty. So meiosis, just like I said, occurs in germ cells, and during meiosis, the genetic material of a diploid germ cell, which is the parent cell, undergoes two nuclear division. But don't forget, the DNA only replicates once, while the nucleus divides how many times? Twice, resulting in four haploid daughters, unlike mitosis, which results in two diploid daughter cells. So meiosis results in four haploid daughter cells, which means the daughter cell will have half the number of chromosomes as the parent cell, and they are not identical. So the stages of meiosis are two. We have meiosis I and meiosis II. Meiosis I is called reductional division because it is at the end of this meiosis you find the chromosome number are reduced to half of the previous of the parent's number of chromosomes. So that is reductional division. While meiosis II is called equational division, just like mitosis. So meiosis II is just like mitosis, which is also equational division. All right. So the first stage of meiosis, just like you always know, the first stage of all cell division, the cell cycle, is interphase. And don't forget the interphase. Just even though we do call the interphase the resting stage, we do say that the cell is not actually resting. because at this stage this is the stage where the cell spends most of its life. And he said at this stage the cell is preparing for cell division. So it consists of G1 phase, the S phase, and the G2 phase. In the G1 phase, we have, which is the growth. So at that phase, the cell is actively absorbing nutrients and growing. Then we move to the S phase. At the S phase, during the S phase, you see the chromosomes that are present here in form of chromatin thread. They replicate. So that's the only thing that happens. It replicates. Do you understand? And then formation of centrioles in animal cells. That's what happened in S phase and interphase. Then the G2 phase also prepares all other organelles that will be needed for mitosis will be formed. Now we'll move to the division proper. Meiosis I consists of four stages, just like mitosis: the prophase, the metaphase, the anaphase, and the telophase. So just like we said, it's prophase 1, metaphase 1, anaphase 1, and telophase 1 for meiosis 1 stage. So it consists of four stages. Prophase 1 is the longest phase in Prophase 1 is the longest phase in meiosis 1, and it is longer than prophase in mitosis. It takes about 90% of the total time for meiosis, and it is the phase that is much more complex than other compared to other stages in meiosis 1. Prophase 1 is divided into 5 stages. We have leptotin, zygotin, pachytin, diplotin, and dikinesis. Don't forget leptotin, zygotin, pachytin, diplotin, and dikinesis. Laissez-faire. So those are the stages of prophase 1. So the first stage, which is the leptotin, or which is also called leptonema. At this phase, don't forget, before the cell enters into division, at interphase, the chromatin granules, the DNA here replicates, and then centrioles were formed. So at the leptotin stage, what will happen in leptotin? The first thing is, just as usual, what will happen in prophase in mitosis? The chromatin granules will coil, condense, and shorten in order to become more visible as chromosomes. And then at this stage, in leptotin stage, there was also increase in nuclear volume, and there is homology search. So what we mean by homology search is homologous chromosomes which are alike and identical in the structure and the arrangement of the genes will actually begin to search for themselves in order to appear in the next stage. So this is what happens in leptotin. The only thing we say happens in leptotin, said what? The chromosomes coil, shorten, and condense in order to become more visible as chromatin chromosomes. That's the chromatin coils, shortens, and condense. Then there is also increase in nuclear volume. Then the next stage is the zygotin stage. So don't forget, after leptotin is zygotin, or zygonema, also known as zygonema. So what happens here is homologous chromosomes pair together. So the pairing of homologous chromosomes, I told you homologous chromosomes are chromosomes that are identical in the arrangement of genes. So the locus, the location of the gene in both chromosomes are identical. So they pair up together. As you can see, this pairs up with this pairs up with this pairs up with this pairs up with this. And the pairing, one will be from the maternal parents, while the other will be from the paternal parent. So that's how we'll have it for the pairing. And that's the only thing that happens in zygotin. So don't forget, leptotin, the chromatin coils, condense, and shortens to become chromosome, while the zygotin or zygonema, pairing up takes place. But don't forget, before pairing up takes place, homology search takes place, which happens in leptotin. Then we come to zygotin, pairing up takes place. So they pair together, and the pairing up is called synapsis. The process of pairing up between homologous chromosomes. Don't forget, it's between homologous chromosomes that are identical. One from the maternal parent, one from the paternal parent. So the process of pairing them up is called synapsis. Another thing happens at this stage, uh, which may not be in the syllabus of the O level, is we have a formation of the synaptonemal complex. So the synaptonemal complex, the function is it helps in pairing up. It helps in pairing up and holding those chromosomes together uh, when they pair up. So when the chromosomes pair together, you can see two chromosomes now. So they are called bivalents. They are called what? Bivalents. But don't forget, a chromosome consists of two sister chromatids joined together at the center, isn't it? A chromosome consists of two sister chromatids. So another chromosome consists of two sister chromatids. So if you join two sister chromatids with another two sister chromatids, how many sister chromatids do you have? Four sister chromatids. So that's why it is called tetrad. So is that you call it bivalent? That's in relation to the chromosome. That's two chromosomes there, or bivalent, which means we have four chromatids that are there. Two sister chromatids and another two sister chromatids. So that's what happens at the zygotine stage or zygonema stage. So the next stage after zygotine stage is what stage? The Parkinson. Don't forget le, ze, pa, di, dia. Le, the leptotene, this zygotine, then the Parkinson or Parkinema. So during the Parkinson stage, one of the most important process in meiosis happens at this stage. So after pairing up in the previous stage, the zygotine stage, the chromosomes, the homologous chromosomes that have paired up at this stage cross one another. So what is called crossing over takes place here. Don't forget the two homologous chromosomes that are lying side by side, that's the bivalents. So we have them like this. So what crossing over takes place? Something like this. You can see them lying side by side, homologous chromosome. These are two sister chromatids. These are also two sister chromatids here. Then they cross over. So a non-sister chromatid with another non-sister chromatid cross over between those two homologous chromosomes. So they cross over in order to exchange genetic material. And the position at which they cross over, the point at which they cross over is called chiasmata. The point at which they cross over is called what? Chiasmata. So they may cross over in one or more points. It can be here and together with here. So it may not be only one point. And it's the exchange genetic material randomly. So the process, this is what is called crossing over, and crossing over is one of the important process in meiosis. The function of crossing over, one of the importance of crossing over is it leads to diverse. So the importance of meiosis, one of the importance of meiosis, it leads to the importance of crossing over is that it leads to increased genetic variations among offspring. So that's why no two children from the same parents are genetically identical except for twins. So it leads to it gives rise to increased recombination and then genetic diversity. So that is crossing over. And don't forget, the point at which crossing over takes place is the chiasmata. That's after pairing up has taken place in the zygotine stage. And then the stage in which crossing over takes place is the Parkinson stage of Prophase 1. Don't forget that. So after Parkinson stage, after crossing over, the next stage is what? Diplotene or the diplonema. So at this stage, the recombination or the crossing over is already complete. You understand? The crossing over is already complete. So as you can see, it's the chromosomes are still there, and at this point it's still called tetra, just like I explained the other time. And then or the synaptonemal complex that we said formed in zygotine stage breaks down because you don't need to pair again. they've already completed what the process of what they want to do, which is the crossing over that happens in packaging stage. So in diplotin, so at this stage, the diplotin stage, after packaging, after crossing over in packaging, so they've exchanged material, the recombination is complete, then homologous chromosomes begin to move apart. They pull apart. They no longer stay together. They no longer pair up again. So that's what happens in the diplotin stage. So first thing I said, the serotonermal complex begins to break down. Then the chromatid, that's the sister chromatid of each homologous chromosome, continues to shorten and thicken again, and the paired chromatids, they begin to pull apart. The paired sister or homologous chromosomes move apart. So that is what happens in the diplotin stage. Then we move to the last stage of prophase, which is diakinesis. In diakinesis, nucleolus disappears or disintegrates. Nuclear envelope also disintegrates, and then the centrioles migrate to the opposite pole of the cell. Then spindle fibers are formed. So that is what happens in diakinesis. So don't forget, these are all everything that this is the completion of what happens in prophase, just like you learned in mitosis. That's in prophase, the nucleolus and the nuclear membrane and the nuclear membrane will disappear, the nucleolus disappears, the spindle fiber is formed, and the centrioles migrate to the other poles of the cell. So after the pairing up or whatever it is that happens in the stage, and then the moving apart in zygote in the packaging stage, and the moving apart of the homologous chromosome in diplotin stage, so the rest of what will happen in prophase happens in diakinesis. So after that, the cell moves to metaphase. As you can see in metaphase, just like other metaphase, the homologous chromosomes arrange themselves in the metaphase plates, that's at equator. But unlike the metaphase in mitosis, where it is only one chromosome that you see, which is attached at both sides by the spindle fiber, at this stage, um, the homologous chromosomes, one is attached to the other spindle fiber from the other end, while the other is attached to the other spindle fiber from the opposite end, after arranging themselves in the metaphase plates or the equator. So that is what happens in metaphase. As you can see, it is arranged. So after that, we move to the next stage, which is anaphase. At anaphase, the homologous chromosomes are pulled apart. In anaphase, they are pulled apart to the opposite pole. They are pulled apart to the opposite pole of the cell. So that's what happens in anaphase. They are pulled apart to the opposite poles of the cell. Don't forget, in metaphase, after whatever it is that happened in prophase, though in metaphase, the homologous chromosomes, which are already having new combination of genes, now arrange themselves at the equator, which is the metaphase plate. After that, in anaphase, they are being pulled to the opposite pole of the cell. One great difference between the anaphase of mitosis and anaphase of meiosis is that in the anaphase of mitosis, the sister chromatids do not remain together because they are being pulled apart to the opposite poles. That's for mitosis. But in the anaphase of meiosis, the anaphase 1 of meiosis 1, the sister chromatids remain joined even after being separate, even after being drawn to the opposite pole. Whereas in mitosis, they separate. So at this point, the chromosome number reduces from 2n, which is the diploid, to n, which is the haploid. After that, in anaphase, we move to the next phase, which is the telophase. So at telophase, it has already the pooled homologous chromosomes have already reached the opposite pole of the cell. So the nuclear envelope comes back, reappears. The nucleolus also reappears, and that is the end of meiosis I. And then, well, don't forget, we have that's not the end. After telophase, that's the end of the main division. So the nucleolus is back, the spindle fiber it disintegrates, then the nucleolus reappears, the nuclear envelope also reappears, and then we have the next one, which is cytokinesis, which is the formation of cleavage furrow here in order to divide the cell into two. So that's cytokinesis. That happens at the end of telophase 1. So now we have two haploid daughter cells at the end of what? At the end of meiosis 1, we have two daughter haploid cells. So after the formation of these, we move to the next one, which is meiosis 2. So the cell enters the next phase, which is meiosis 2. In meiosis 2, just like I said, is similar to mitosis. So it's just an equational division. So what happened? We have the prophase. And the prophase, what the difference between meiosis 2 and mitosis is that we don't have any other DNA replication. DNA does not replicate again. So just like I said in the beginning, that in meiosis, during meiosis, DNA only replicates once, but the nuclear division takes place twice. At prophase 2, each chromosome is called a dyad, and it's composed of a pair of sister chromatids that are attached together at the centromere, at the center. Then in metaphase, what's just like in metaphase of mitosis, the chromosomes are arrange themselves at the equatorial plane, at the equator, at the metaphase splits. That's all what happens at metaphase 2. Then anaphase 2, same thing. What happens at in the anaphase of mitosis, the chromosomes are pulled apart at their center. So the centromere, which is the center that is holding the sister chromatids together, divides, and each sister chromatid will divide to become daughter chromosomes and are pulled apart. So just like unlike the anaphase 1 of meiosis 1, in which the sister chromatids remain joined, the anaphase 2, the sister chromatids are what separates, and that's what happens in anaphase 2. So move to telophase 2. The daughter chromosomes, that's after the sister chromatids are being, after, we have the chromosome consists of two sister chromatids. So once they are pulled away together, once they are pulled apart, they are called, each one is called daughter chromosome. So the daughter chromosome have reached the end at telophase, they've reached the end of the pull. So what will happen is the chromosome will lengthen, uncoil to become chromatin thread. It will lengthen to uncoil to become chromatin thread. That's a telophase. Then the nuclear envelope reappears, the nucleolus also reappears, and cytokinesis takes place. Then the next phase, the last, after there's a telophase. Then whatever it is takes place at telophase. Then we move to the last phase, which is cytokinesis. So cytokinesis takes place, and then we have four haploid cells after the meiosis 2. So we have four haploid cells. So that's the end of meiosis. Then the functions of meiosis: one, production of gametes. So haploid gametes, don't forget haploid gametes. So what will happen is during fertilization, the sperm, which is having haploid gametes, the egg is also having haploid gametes. When they fuse together to form zygotes, they restore the number of chromosomes of chromosomes to become diploid. So haploid plus haploid, they will come back to be diploid. So another function of meiosis is production of tremendous amounts of genetic variation. So one of the main function of meiosis is that it is essential for continuity of generation, because that is what produces the gametes, and it is only the union of those gametes that will lead to development of new individual, lead to development of zygotes, thereby forming a new individual. So meiosis is essential for the continuity of generation, one of the most important things. Now let's try to check the differences between some little differences between mitosis and meiosis. Mitosis occurs in somatic cells, while meiosis occurs in reproductive cells, germ cells. Mitosis produces two daughter cells. One cell will produce two daughter cells, while in meiosis, one cell will produce four daughter cells. Mitosis is an equational division separating sister chromatid. But meiosis, don't forget, we have meiosis I, which is reductional division, while meiosis II is equational division. Do you understand? So in the meiosis I, which is a reductional division, it just separates homologous chromosomes, and sister chromatids are not separated. Sister chromatids only separate in meiosis II, which is the equational division. So don't forget, mitosis is just equational division, while meiosis consists of reductional division, which is meiosis I, and meiosis II, equational division. Then in mitosis, only one division per cycle. That's one cytoplasmic division and one nuclear division per equational division. But in meiosis, DNA only replicates once as in mitosis too, but the division is what? Twice. So two divisions per cycle, two cytoplasmic division, two nuclear division. And those two division, one is reduction division and the other is equation division. Which one is reduction division? Meiosis one. And the equation division, which resembles mitosis, is meiosis two. In mitosis, there is nothing like synapses. There is nothing like chromosomes pairing up, and no chiasmata is formed for crossing over. But that happens in meiosis. Crossing over takes place, and another one is genetic exchange between homologous chromosomes. Does not take place in mitosis, but takes place in meiosis during what? During crossing over. In which stage? In packaging stage. Another one is genetic contexts of daughter cells are identical. So mitosis produce two daughter cells that are identical, but meiosis produces four daughter cells that are not identical. So that's how we have and the main reasons why they are not identical is because of the crossing over that takes place in packaging stage. Another one, chromosome number of daughter cell is the same as the mother cell, so it produces diploid cell, but meiosis produces haploid cell, so the daughter cells having half number of chromosomes as the mother cells. You understand? So another difference between mitosis and meiosis is that the daughter cells that are formed in mitosis after growing are capable of undergoing another additional mitotic division, while the daughter cells that are produced, the haploid daughter cells, that's the gametes, that are produced from meiosis, do not undergo meiotic division. Don't forget, the gametes do not undergo meiotic division. It is the germ cells that undergo meiotic division to form the gametes. So from there, what will just happen is the gametes may undergo mitotic division though, but not meiotic division. So, and that happens during fertilization. For mitosis, it starts at zygote stage, that's immediately after fertilization, to form zygote, to form a single cell. So the zygote, the mitosis starts from there to form two cells. It takes to form four cells again, to form eight cells, and continues to divide using mitosis, and it continues throughout the life of an organism. Don't forget, we said mitosis is for growth, for repair of worn out tissue. So, and growth and repair of worn out tissue happens throughout the life of an organism, and also asexual reproduction in some organisms that undergo asexual reproduction. But meiosis, just like I explained in the beginning, only occurs after puberty in high organism. Occurs only after puberty. But he said meiosis starts if it is in a female. It's that's oogenesis. It starts when the female is the organism is in the mother's womb. So this is a diagram showing meiosis in higher organism. So he said meiosis is the formation of gametes. That's gametogenesis. In the male, it is called spermatogenesis, while in the female, it is called oogenesis. So you can see after meiosis one, we have two haploid cells. Two haploid cells. After meiosis one, we have two haploid cells too. That's here two haploid cells that will later become sperm, then two haploid cells that will later become um, egg. But unlike the meiosis in the male, the meiosis in the female, after producing two daughter cells, two haploid cells, one become a polar body. Then after meiosis II, we have four haploid cells here, and we also have what? We also have four haploid cells, but only one differentiates to form a single egg, while the other three becomes polar bodies. So unlike the spermatogenesis, where all the four haploid cells become spermatozoa. So that was the end of meiosis. If you have any question, so let's recap meiosis again. Just like we said meiosis in male is called spermatogenesis and it produces sperms, while meiosis in female is called oogenesis and it produces ova. We have two basic division, meiosis I and meiosis II. Meiosis I is a reduction division, while meiosis II is equation division. And we have it like this, where mitosis produces only somatic cells that are, so you see the parent cell, we have two daughter, two, and which is diploid, also produces diploid. But for meiosis, it's two cell division and produces four haploid cells, having half the number of chromosomes as the mother cells. The other thing, definitions of some terminologies. Homologous chromosomes, just like we explained, homologous chromosomes are pairs of chromosomes that are identical in the arrangement of genes. One will be from the maternal parent, the other will be from the paternal parents, and they pair up in which stage? They pair up in the zygote stage, and then crossing over takes place in Purkinje stage. Don't forget that. So we have the synapses. The synapses, that's the pairing up that takes place where? In which stage? So synapses is the pairing up of homologous chromosomes that takes place in the zygote stage. Don't forget that. So synapses is the pairing up that takes place. Then we have the crossing over, al-ubur. The crossing over takes place in which stage? In the Purkinje stage. And that leads to the diverse genetic variation. So crossing over takes place is the process of exchanging genetic material between two non-sister chromatid. Don't forget, two non-sister chromatid. And they cross over and we have the recombinant chromosomes. Then we explained the meiosis I, which is the reduction division, said the longest phase and is divided into leptotin, zygote, Purkinje, diplotin, and diakinesis. And this is it in leptotin. You see what happened? The formation of the movements of the condensation that takes place, the chromatin granules condense and coil and shortens to become chromosomes. And at the zygote stage, pairing up takes place, that's synapses. In the Purkinje stage, crossing over takes place, and in the diplotin stage, the what's it called? The centrioles move to the opposite pole, and then we have the separation of the crossing over is complete. Then we have the diakinesis, where the nucleolus disappears, the nuclear envelope also disappears, and it moves to the next phase, which is metaphase. So at metaphase, they arrange themselves at the equator. At anaphase, they are being separated, even though the sister chromatid do not separate. At telophase, the nucleolus reappears, the nuclear envelope reappears, and then cytokinesis starts. Then after cytokinesis, we'll move to prophase 2. Then after prophase 2, we have the metaphase 2. This happens at metaphase 2. The homologous chromosomes arrange at the equator, and at anaphase, they are being separated. But this time around, anaphase 2 of meiosis 2, the sister chromatids are also separated, and once they are separated, I said they are called daughter chromosomes. At telophase, the nucleolus reappears, the nuclear envelope also reappears, and then the spindle fiber disintegrates.